I'm here to provide a CDMO perspective and seeing what AAV manufacturing challenges we have as a CDMO and where the role of AI would absolutely support this. Little background about resilience. So we actually service multiple modalities, biologics, vaccines, nucleic acids, cell therapy, and gene therapy. And our goal at Resilience is not just to be your CDMO, but really implement novel technologies and novel platforms to really get your therapies to the clinic and faster and lower costs. But that is, we're working with our biotech partners to be able to bring that in. We work with leading research institutes. I'll show a list of few folks we've partnered with, large pharma, and we actually also have a government arm. So right now, is that we're 5 modalities. We're working on over 70 plus molecules, and we're growing from development all the way to clinical to commercial scale. We work with research institutes, but most of our partners are, as you see, are our biotechs, people like Solid Bio, people who are smaller biotechs that look for partners in the manufacturing space to get their products to, first in human trials or in further. Some of our partners are large pharma, and I said we have a government arm. We do a lot of that work down in our Alachua facility in Florida. Some of the partners we have, you might recognize a lot of these partners. It's not just academia, but pointing out Bridge Bio and B Bio, those are biotech in the space. So those are public announcements that we have out there that we're working on their products with them. And then we partner with our academia academics and the PIs there as well, because they're helping drive some of the science. So they, us being able to provide that first in human work for them helps them to get quicker results in the clinic. So this is where we are in North America. We have multiple manufacturing facilities and some development facilities. So I'm based out of Boston. So we have a facility. It's the old Genzyme building, Sanofi, near the Alston. You can see it from the Charles River. It's a beautiful facility where we do most of our development work. But we have other areas. And then on the West Coast, we actually have more advanced research places in San Diego and the Bay Area. Where we do a lot of our manufacturing for vector is actually at our RTP facility, got commercially licensed earlier this year, and we actually have a commercial product there. But that's the facility where we are taking it all the way through. We can get up to 2,000 liters if needed. Right now we have experience going up to 500, but we can expand up to 2,000 liters if needed. So I'm just showing you all this. This is a network we built to really support the different modalities and especially gene therapy. So what are we trying to do? So we want to get your therapy to the clinic. We want to get you the accelerated path to IND by giving you, great platforms, innovative technology that gets you there. We are addressing the capacity challenges across the industry. So we have experienced teams. So the facility that we have in RTP, we actually acquired from Bluebird about a few years ago. And with that came with a lot of knowledge and expertise. Those folks are still there. So while we might only have been owners of them for about 3 years, we do have a lot of the knowledge and expertise of those folks who've been there for many years before that. And the capacity, plenty of capacity. I mean, we bought these facilities to answer the challenges that there's a backlog in manufacturing LVV for these cell therapies. So there you go, we have some space there. And then with the commercial manufacturing experience, I just mentioned to you, we're a commercially licensed facility. So with that being said, we now can manufacture on the commercial product there, both AAV and lenti suspension. So the platform that I mentioned, that we have a suspension platform. Apologies to the animations here, I didn't see that. Oops. All right, I hope you guys can see that. I think it's grayed out at the top. I didn't realize that. But it's your standard triple plasmid transfection process. We've engineered this and developed this process on our own. This is our own Resilience own process. And typically, we're taking your bench scale process and taking it all the way to a larger scale process so that you can get to the clinic and commercial scale. So cell bank to shake flasks to bioreactors, and then multiple different engineering steps so that we can purify this, clarify this, and package this in vials so we have drug product filling down RTP as well. We have a whole slew of analytics. We developed the analytics up here in our Alston facility, and those are transferred down to our RTP facility. So process development and resilience. So we have this whole process, but what are we trying to do? We want to see the candidate that you're bringing in fits within our process, right? We want to make sure there's a platform fit. So what we typically do is start with a smaller scale, so maybe 3 to 10 liters bioreactor. We're going to let you know, hey, this is the output that you require, right? So we are going to assume a linear scale up to 200, 500, wherever you have. So if it comes out here, it's going to come out to that output at that larger scale. Then if needed, we can do some process optimization if you are not satisfied with what comes out of that platform fit run. We have knowledgeable folks within our team that can help optimize this. Is it plasmid ratios? Is it seeding density? Is it the timing? There's a lot of DOE that we can do in-house to optimize your process. And then we will do some shakedown runs. We'll make sure it all works. And then we'll scale it up. And at our Alston facility, we actually have a pilot lab. So we can do a demo run for you and show you that at the 50 liter or 200 liter scale, this is what you're going to get. And the way we've set it up is that we can actually do this as make tox material for your preclinical work as well. So as a CDMO, we're trying to set up what we can at our development facility so that we can de-risk the transfer and de-risk the manufacturing process down at our RTP facility. Everything we have here is replicated down there. And that's typically what a CDMO wants to do, is showcase to the customers that we have all the knobs that we can turn. We have all the bells and whistles. We have everything we can do to make sure you see success in your manufacturing and you can get your IND filed. So with that being said, we've done a lot of runs within Resilience. This is our titers versus our full ratio across, different scales and different serotypes. And you can see we're working on a lot of the common serotypes here, 2, 8, and 9. If you look at a market analysis, those are the most common serotypes folks are working on. So we've definitely seen a lot of customers coming in and giving us that information. And we also have a lot of experience with our AAV 6 and 7 as well. All right, so this is the data. Great, we have this. But do you see those lines going up and down? There's definitely variability, right? There's variability across the serotypes and variability within the serotypes. Why within the same serotype are we seeing these variabilities? Biotech, someone like Solid Bio or B Bio, they're coming to us saying, you have a platform process. Put it in and give me something out. Every time it should be the same thing, right? If I gave you an AAV6 for this construct, the AAV6 for another construct, that's what you're supposed to give me, right? But how are we as a CDMO trying to address that? We say we can optimize this, the transcription ratios, your cell seeding density. We can engineer the hell out of it and tell you this is what we're going to give you, right? But what if the GOI that's causing the issue, right? How can we go back to you and tell you what you're giving us, your baby, the one you've spent multiple years, lots of money and resources, and saying your GOI is causing the issue? Regardless of us getting to a 2,000 liter batch, you are still not going to get what you need to dose your patients and your population. So our only suggestion to you is let's go and do a resupply. That's what we can do as an organization, right? As a CDMO, we do not have the upstream portion of this where we go re-engineer or develop your GOI and design for you. What we do is take what you give us and try to make the best of it from our side. We have a whole team from our development team, our MSAT team to manufacturing that's going to put all the blood, sweat, and tears to make sure that we see success in the clinic. So that's usually not the favorite answer that our clients like when they said you need to spend more time and money and get another batch, right? But this is the challenge that we're facing. So usually clients, as I said, come to us. They've already down selected their candidates. And that's what CDMOs do. We intake what you give us, and now we're doing the process development to scale up your process. We're not going upstream of that and really developing our construct. Sometimes we need additional confirmation to demonstrate the platform fit. We've had clients come to us saying, hey, I have 3 candidates. I don't know what to choose. Can we just put in your platform and make sure it works? And we'll go from there. And we were having a conversation with Form Bio, I think a few weeks ago, and Mark actually mentioned this. When the manufacturing fails and you're not getting what you want at the end, is it a design problem or is it a process problem? The process problem we can address, but we can't address the design problem. And at this point, we might be too late. And depending, we have to go back and re-engineer everything, and now it takes more time and money. So what are our clients doing? They're finalizing the lead candidate based on data either they're generating, but what if two constructs give you the same data? You're not going to go back to your technical team or your board and saying, hey, we flipped a coin and this is the construct we're going with because, you know, it just works. Because you downstream might have to justify why you made that decision. So that's an issue that we see. We actually had a client come in with 5 constructs. And going through 5 constructs and doing that down selection took about 3 to 4 months to make sure that we're getting to the right data. But that puts more pressure on them and to choose something, because at the end of the day, a lot of our clients, are small companies. They're not public. They're private companies that have a little bit of funding, and they don't have much to spend to move this forward. There's timeline and resource constraints. A lot of these clients need to get to the first in human by X months to make sure they unlock the next level of funding. I talked about low production that's leading to multiple batches for IND and phase 1 trials. We currently do have clients that are not satisfied with the output that they're getting, even from our scale-up run at 50 liters. And, they believe that, but I asked for this, you're supposed to give me this. But, it's hard for us to have that conversation with them saying, it might just be your gene of interest. It's your construct design that we need to go back and look at. And then once you start filing your IND, you're locked into the IND, locked into the construct, right? Going into later phases, and we're scaling up to, for phase 2, pivotal or commercial, at the end of the day, you just have to say, I need 6 batches to get to my clinical trial. I need to get 6 batches to even get a commercial supply. That's what our clients are facing at this time. So how do we address this? As a CDMO, as I said, we can only tune that to get to what we need. So how do we partner with folks like Forum Bio to provide these solutions to our clients? Well, as I said, a lot of people come to us already with their construct selected. But is there a way to just verify this before we get deep into manufacturing? I believe it was mentioned it could be a quick turnaround time to just take a quick look. So we get those in silico methods to screen and select GOIs that promote better capsid yield and quality. Let's do a construct review. Even before, maybe it's in parallel to the down selection we're doing in-house, we just do a construct review, just so if we do end up getting those 2 constructs that do look pretty identical from, our PE runs, that now we could just look at the data from doing a construct review and making a more educated decision of which one we choose. We're not flipping a coin. Data-driven decision-making is what really drives success. And then at the end of the day, we have plenty of companies that have pipelines. If we're not able to implement this with their first product, let's work with that company to show the benefit of how this could, help see more success in the clinic and your manufacturing by showing how you can do construct design to reduce your time to lead candidate and ultimately to the clinic. Now, this is very general, right? We're talking about AAV here, but I see this as an expansion into multiple areas where people are having trouble with titers and innovating in that space. We see this issue with LVV. LVV, similar to AAV vector design, there's designs that we are observing that have better production within our platforms versus what our clients see on their own bench scale. Biologics. Biologics hasn't really, biologics, we all know how to manufacture it, right? But there's definitely a lead selection, a candidate selection process that is very much needed. And there are people out there innovating on this, but we need to find ways to, once again, take from 1 million candidates down to that particular 1 or 2 candidates and showcase success. Because the more that you get out of your end yields is definitely going to reduce your COGS and your batches and just get more people the medicines. So to demonstrate some of this, right? We've only done very small scale partnership with Forum Bio, but it's really very valuable of what we did see with this case study. I'm not showcasing all the data at the case study. If you'd like the case study, Forum Bio has a copy. We have copies too, happy to share, and please connect with me. But we basically partnered with Forum Bio and said, let's go very basic. Let's use our model construct, our model molecule, HGH, and see if we optimize it using your algorithms, can we see an improvement just in our basic platform? And We did. So Foreign Bio, we worked with them and they optimized our construct, and they recommended an alternate promoter and sequence optimization. And we saw some, and this was an N01, mind you, but it's still very promising. So we saw an up to a 2-fold increase in genomic titer, which we saw when we used the optimized HES coding sequence, and when also coupled with a better performing jet promoter. We saw improvements in the genomic titer, and basically we were able to package more full genomes using this optimized construct. So as I said, this is an N01, it's a small study, but it's still very promising that even with our model construct that we use to optimize our own platforms, we're receiving slight increased improvements. So these are the kinds of methodologies or opportunities that we'd have with companies to partner and really take a look at what construct you're providing to us. So downstream, after we've spent the millions of dollars, or we've asked companies to spend another $1 million for like another batch, we say, oh, it's a GOI issue. Let's try to find a way, work together, and make more awareness that that construct design is really going to call out how successful you could be in the future. And for a CDMO, if you come in with a better designed construct, we can definitely show more success. And that's exactly what we have seen with some of our other clients. So with that, I do want to thank Form Bio for working on that work with us.